Hello, class, and welcome to chapter 10. This chapter will be on the structure and function of DNA, and this can be a really lengthy chapter. And you'll notice I try to keep my lectures a little bit simpler to focus on what I want you guys to learn. In this chapter, I've also included a few video links underneath the chapter PowerPoint in Canvas. So please take a look at those animated video links because they take you through the structure and function of DNA. We'll go over a little bit here about the structure of DNA, and you can read through this, and please, watch these animations embedded within the PowerPoint as well. But the typical structure of DNA is made up of a phosphate group, a sugar, and a nitrogenous base. And you have four different nitrogenous bases: adenine, cytosine, thymine, and guanine. And they will be able to kind of complement each other when the double strands of DNA will be formed. RNA is a little bit different because instead of a thymine nitrogenous base, it will have a uracil. So this is a look at an RNA nucleotide, and we'll talk about the differences between each. So Watson and Crick were the ones who kind of discovered this structure of DNA, looking at kind of like how it looks like a full ladder twisted up into a spiral, where the sides represent the sugar phosphate backbones, and each rung in DNA represents a pair of bases connected by hydrogen bonds. And the base pairing rules will dictate the side-by-side combinations of bases that form the rungs of the double helix. So you can kind of see here the double helix structure of DNA is like a ladder with a twist. And we'll kind of continue on looking at how this ladder is formed. We have a ribbon model showing the hydrogen bonds. Then we have the atomic model and the computer model. This atomic and ribbon model are showing how the nitrogenous bases will be complementary paired up, and they will be held together in order to make that ladder-like structure. So that's a little bit about DNA. We know when we talk about cells dividing, their DNA first needs to replicate. And this idea of how cells divide will be really helpful when you watch this animation. But DNA must be replicated before the cell can divide. And really simply, we have a parent, the old DNA molecule that kind of gets unzipped, and then new daughter strands will get complementary paired up with half of that old strand of the parent molecule. And again, there just are some great animations here that I've embedded into the PowerPoint that I would like you guys to watch. DNA polymerases are the enzymes that make the covalent bonds between the nucleotides of a new DNA strand and help repair DNA that would be damaged by toxic chemicals or high energy radiation. So we have the parental strands. You have an origin of replication, which creates a bubble where new daughter strands form. So at the end of the day, we get two new daughter DNA molecules. And again, origins of replication show where that replication starts in the parent strand. The leading strand talks about how one strand will lead and the others will follow. From DNA to RNA to protein, then, this talks about the sequence of how we go from the instructions that DNA provides to a cell and an organism as a whole. The organism's genotype, or the genetic makeup, is the heritable information contained in the sequence of nucleotide bases in its DNA, and the phenotype, the organism's physical traits, will arise from the actions of a wide variety of proteins. So DNA specifies the synthesis of proteins in transcription, which is when it transfers genetic information from DNA into an RNA molecule, and then translation, which is the transfer of information from RNA into a polypeptide or a protein strand. So the function of DNA is to dictate the production of the polypeptide, and then the relationship between genes and proteins is thus one of information flow. And I really like this picture because this simply shows how we get from DNA and how we create proteins, because DNA holds kind of the instructions that will get transcribed into an RNA molecule that occurs inside the nucleus of a cell. The RNA molecule then leaves the nucleus, enters the cytoplasm, where translation occurs, where we translate those instructions from an RNA molecule to create a protein. So that is, simply put, this idea of transcription and translation. There's a transcription of DNA and the translation of codons. where we have a strand of DNA, and then we'll have an RNA strand that forms, which will create complementary base pairs using uracil instead of thymine to connect to the adenine base pair. And then every 3 base pairs creates a codon, and that codon will code for a specific amino acid that will create the polypeptide or the protein. And again, this is the very simply how proteins are created. The dictionary of the genetic code, those kind of 3 packs of RNA codons will code for a specific amino acid. And we have 20 different types of amino acids, and they'll all be combined in different ways to create proteins. So that is a simple look at transcription and translation of DNA to create proteins. I would like you to look through the PowerPoint and watch these animations and work through your assigned reading for this chapter. So you'll work through that worksheet as well as watching those 2 links. And that is kind of the key points of what I want you to remember from this chapter. Thank you for listening, everyone, and please let me know if you have any questions.